All right, we are live. Hi, and welcome to our Friday webinar. Our special topic today is Avian Bornavirus with Dr. Susan Orosz. She's going to talk about the common signs and treatments of this virus that we should all be well aware of. So welcome, Dr. Orosz. Nice to see you.
Nice to see you. I don't know, can people see me now?
I think so. I think they can. And just real quick, that's your farm in the background, right? That your back
That's
shot
right.
there?
Yep.
That's awesome. It's a beautiful shot. Well, I know that I just seen the numbers here. This is a topic that is a lot of people are logging on for. So that's very exciting to see. And I know you got a presentation for us today. So just a couple things before we get started as we wait for people to log in. I'm going to play a video from our sponsor, which is the Lafeber. We're doing the Fruit Delight Avi cakes, and this is if you stay tuned to the end of the webinar, because we'll be giving some Avi cakes away, as well as some other Lafeber product to one of our our giveaway prize today to one of our viewers. So let's do that, and we'll let people log in, and then we're going to turn it over to you, Susan, so you can get.
Great.
Okay, here we go. So I'm going to do a screen share. Screen shares are always cross my fingers no matter what, just because screen shares are fun.
Okay, so am I screen sharing now, or are you showing the video?
I'm going to show a video real quick, and then when I'm done with the video, I'll just make a couple quick announcements, and then we'll have you roll.
All right. Okay, great.
Here we go. So now I am screen sharing. Let's see. Okay. All right, hopefully people can see that. Okay, so Dr. Orosz, this is also this is one of Lafeber's top products, and I know that you have had a big part of developing a lot of the diets for Lafeber, such as the senior nutriberries and stuff. So this is someone who's put a lot of thought into these diets as well. Make sure that birds are eating healthy.
Yes. As we all know, Dr. Kurt Claesing from UC Davis is the nutritional brain behind a lot of this, but it's very exciting. Particularly, it took me about, I think, 20 years to get Ted to decide to do a senior nutriberry.
Oh, nice. Because birds are living longer, so we want to give them that nutritional support, right, as they age.
Right, exactly.
Nice. All right, there we go.
Okay.
And then just real quick, a couple announcements. We're not going to do a Q&A today, but if you do have a question about the topic, Avian Bornavirus, you can submit that to us, and then we're planning on having a follow-up webinar in October with Dr. Orosz, and as well as Dr. Dahlhausen. So with that being said, I'm going to turn it over to you, and welcome to the webinar. If you're just arriving, we are going to be talking about Avian Bornavirus today with Dr. Susan Orosz. Oh, there we go. You can see your screen perfectly.
Okay. So I want to say that this has been something that I've been really interested in for a really long time. So, Laura, everybody can see this now? Can they see this?
I believe so, yeah.
Okay, so in my first life, I was a professor at the University of Cincinnati Medical Center, and I was a neuroanatomist. And after that, I went to vet school at the Ohio State University. And my first stint out was to work in San Diego, California. And this little bird here in this blue and gold macaw was one of the first photographs. This is a historical photograph of a bird that had this undigested seed in its feces and subsequently died. And at the time, all we knew to call it was macaw wasting disease. We had no idea what this was. This is one of the first descriptions of it. There's some other people, Dr. Gerlach and others, that were about the same time writing about this. And then on necropsies, we noticed that these birds also had dilation of the proventriculus. And from a neurologic perspective, as we understood a little bit more of it, neurologically, this is really a disease of the nervous system, the central nervous system, as well as the peripheral nervous system. And now we're calling it avian Bornavirus disease. And what I'd like to talk to you about today is to think about it in a broader scope. And can we have the disease without avian Bornavirus? And when we look at it, we have a variety of descriptions of this disease. And I think to reduce that problem, I think it's better to describe it as Avian Ganglioneuritis. So often I'll call it AGN, because this is a disease when you actually see symptoms or signs that depending if you're the clinician or the owner, you see things that relate to the nervous system. And we're going to talk about that today. So the classic disease as we saw it was that of the gastrointestinal tract. And I think my pointer's working, and you see this very large proventriculus, and it's also very thin. And for those of you who are veterinarians, the first clue is the thinning of the GI wall, and that's when we have neurogenic atrophy. Okay, so at this point, as a neuro person, I said, oh gosh, the nerves can't be working correctly because the wall is getting thinner. So we found that we could describe this disease typically in New World species, and the classical presentations were things that relate to the GI tract. And if you looked at it more closely, it was the proximal part of the GI tract, which means the upper part or the beginning of the GI tract, which included, sorry, I got my cat in, things that involve the crop, the stomach, which is the proventriculus and the ventriculus. And so associated with that is birds who got depressed, they didn't want to eat. If they did eat, they had undigested products in their feces, typically back then. In 1984, when I was first looking at this disease process, I used to have a lot of seed because that's what we had back then. And then associated with it, loss of body condition. Certainly birds would have delayed crop emptying, or they would regurgitate. And so we had all these different things that we saw. Okay, and here you see this was a green-winged macaw with severe muscle wasting associated with it too. And one of the things that I said on the front end was that it involved this part of the nervous system, the vagus nerve, which is the 10th cranial nerve of the mammal and of the bird, and it supplies nerve supply to the esophagus, the crop, the distal esophagus, and the proventriculus, the stomach, and the first part of the small intestines. And so you could have problems with the crop, you could have problems with the proventriculus, you could have problems with all of it or a little bit of it. And this shows you an enlarged proventriculus. Here's the gizzard or the ventriculus. And on a radiograph, you see here's the heart. The head is this way, the tail this way, a very enlarged crop. And then you see a very dilated proventriculus, and then the ventriculus is down here. So this could happen in any or part of it. And this involves the vagus nerve. And the vagus nerve, in addition, supplies the heart. So when I said the dorsal vagal nucleus, what I'm talking about is the cells, the cell bodies that are in a part of the brainstem, the back part of the brainstem, which is the medulla. And there's this nucleus there, and that is going to be controlling different things, different organs, which is the heart and the proximal part of the GI tract, from the what I said before, from the esophagus down through the first part of the small intestine. Interestingly, when we talk about, when we add in some ideas about feather picking, we have to think about innervation to the heart. And it seems that we tend to see lesions more on the right side and the left side. And you see this little gray here. And surprisingly, there's more innervation of that vagus nerve to the right side of the heart. When we look at this cerebrum, or the part of the brain that is involved, like here on the right is the human cerebrum, and it has these gyri and sulci, and birds don't. They're lissencephalic, which means they don't have those. And for a long time, we didn't realize that meant birds, we thought because this was smooth, we thought it was smooth, that birds were stupid, right? Bird brains, they're bird brain. And now we're learning that that's a pile of poop. That was all a bunch of junk. And so this area here is the cerebrum. And pathologists also found that in addition to ganglion cells of this outflow from the vagus nerve that are found within the heart or the first part of the GI tract, they also found these other things where we had glial cells, we had some cuffing, all these different problems. We had lymphocytic invasion. These lymphocytes are these smaller cells right here. See, all these things they're zooming into that tissue. I found that, and we found that there were also symptoms sometimes, depending on, which we didn't really understand very well either, in the glia and the granular cell layer of the cerebellum. So here is, down here, here's the bird brain, here's the human brain. And I always like to relate to humans because I was a human neuroanatomist, and then I can explain that from there. But here is that cerebrum, and this area here where you have these, gyri and sulci, this is the cerebellum. And the cerebellum is associated with motor control or smoothness. The cerebrum says, I want to move my hand and I want to pick up my glass. And the game plan of that action comes from the cerebellum. So when you had these disruption and loss of these Purkinje cells, and you had these other changes in the molecular cell layer, which they found with these birds that had PDD, quote unquote, there were things like ataxia, and I'm going to show you this here in a minute, and some proprioceptive deficits, which means the animal did not know where its body was in three-dimensional space. You're with me, I can tell. Okay. Here we have ataxia in coordination. There's this kind of shimmering or shaking because this bird just doesn't know exactly where its wings are. And I'm sure it doesn't know where its feet are either, because have you seen a cockatoo just want to sit around? I mean, hell no, and you can tell was an altered mentation. I don't know if I can run this again. Eyes not bright. We're kind of not moving around. We're kind of moving. We're hardly moving at all. And that's something that happens when there's an alteration of the cerebellum. Okay. And then you also get this incoordinated, I don't know where my feet are, my hands are, if you're a human, wings are if you're a bird, and three-dimensional space as well. And paresis is described as a weakness because the things are not lined up in the brain properly. Let me show you this one. This was a poor little bird that was positive for PDD, or, you know, we learned Avian Bornavirus, and he died shortly after this video. Here I am showing you a postural reaction you're supposed to take and pull the wings out, and they should snap back because the bird should know where the wings are, where his wings are. And I know he's not going to bite me because he can't figure out where his body is. So it's pretty easy to see. Let's do this one again. So you see here, I'm trying to pull his wing out, and where is it? And you kind of see this kind of reverberation sense of it. And he doesn't know where his feet are. So all of that is messed up. And that's something that was happening look at those birds. They were old world species of birds, different than the new world species. And then here we have this problem that we get this kind of petit mal type seizure. In other words, maybe the wing would shake or the leg would shake. And this is something that we also see with this disease process. Bird has not lost consciousness, right? He can't move. He's not able to move his wings. He doesn't move his foot, but he's got this kind of tapping. Some people have described it as toe tapping that's associated with Bornavirus disease. And here's another little guy, and you can see the same thing. We get this shuddering or shaking with this kind of change in the beak and the bill, kind of a shimmering type thing that happens. Again, another bird with avian Bornavirus disease, or avian Ganglioneuritis would probably be a better description. So I wanted you to see some of these things and try to understand them in the context. Maybe as owners, you haven't thought about it in terms of the fact that there are parts of the brain that are being affected. And from a neuro point of view, from a neuroanatomist point of view, when I would watch these things and think about these things, these birds were exhibiting symptoms that were somewhat unique and different, okay? And then here comes another problem. This was a study done by Berhane, and they talked about the thoracolumbar spinal cord, which is this area in here, this thoracolumbar spinal cord. And this is a cross section of part of the spinal cord. And we have, again, a lot of infiltration of lymphocytes. And we have this swelling of the axons. And when they swell, it causes degeneration of the wrapping. And the wrapping is myelin. So those birds can exhibit signs of a neuritis or an inflammation of the nerves. And this could be perceived then as a bird that might be plucking its feathers. So this was a study done by Berhane, and he felt that it contributed to feather destruction and or self-mutilation disorders. There was another study done by Michael Leertz after that noted that about 70% of his birds that had feather destructive behavior were also positive for Bornavirus. Now that doesn't mean that's the only thing, see? So when we look at feather damaging behaviors, what I have always wanted to try to understand is it makes no sense to me that normal birds would want to chew up their feathers or mutilate their bodies. People do it that have certain, and I've talked to psychiatrists about this a lot. And I've shown them video clips of birds doing some of this stuff too. So for me, as a neuro person, I want to look at it in terms of, can there be pain? Can we understand? Our first thing when we're talking to owners, you guys, and you're describing things to us, do we think it's painful as opposed to a behavioral thing? I talked a lot about originally in 2013 about feather damaging behavior and the behavioral aspects of it. Mm-hmm. But I've been searching to understand this in great detail. And with avian ganglioneuritis, that can be one cause of feather damaging behavior. And I think it's creating, because I wish I could talk to the birds, because I would have learned a lot more faster, right? And so I think there's some change of sensation. Could be pain, could be tingling, could be something. Just like I use the example of diabetic neuropathy. The people with diabetes have alteration of their ability to feel parts of their body. And as we know in birds, when something isn't right and they're close to being from the wild, if they were a wild bird, they want to get rid of it, right? That's what their decision is. I got to get rid of it if I'm going to try to live. And so when they do that, they chew it off. Like I've seen birds chew their wings off, right? And they die when they hit the blood vessel. But so there's chewing and there's all these things. And so we all of us have to look at this very carefully. Other reasons, and I've been working with Dr. Scott Eccles on this, and I don't have a CT, but I promise you, in our thing in October, by then he was flying around today, so I couldn't get any pictures showing. But he's doing a lot of CT studies. Mm-hmm. And we have to think of other reasons for pain. Cardiovascular disease can result in there's something called referred pain, that there's pain, just like when they talk about men with heart disease that are having a heart attack. They're having a heart attack, but they have pain in their left arm. That's a referred pain thing. So that can be happening. And we're going to show you this pinching at the intervertebral foramen, and I'll show you a picture of that. And we talked with Dr. Marshall, for those of you who listened to Dr. Marshall this past Sunday, and he talked about GI pain, GI referred pain, so that there were certain locations that eclectus pluck related to pain in certain parts of their GI tract. So that's why when we talk about feather damaging behavior, we need you to be sleuths as owners. You need to really look at, when is the bird plucking, what time of day, what time of year, is it all the time, is it a few times, is it what happens immediately preceding the plucking? Those are all clues for a diagnostic workup. Okay. And from Dr. Eccles' perspective, because he's the CT genius, right? You guys all have seen his fabulous stuff. I mean, it's just amazing. It's just fabulous. How else can you even describe it? Yeah. So we have to think about that diagnostic workup. We should include Bornavirus in this thing. And we also should look at, is there cardiovascular referred pain? And then the reason I'm showing you this is, here's a vertebral column. And he has seen in many instances, this little hole right here is where the nerve shoots out, the intervertebral foramen or foramina. Multiple ones are foramina. And that's where the nerves exit. And this is a whole other area. Laura will have to talk about flight sometime, yeah. And how fabulous flight is, okay? So when birds don't fly and they sit on their butts all day, they start to lose bone quality. And when they do that, this thing can collapse down and put pressure on that nerve. And that can cause that pain. And then the bird plucks because that nerve innervates the body wall at a certain spot, right? And that can be times when you have pain. Okay,
Oh,
So,
Dr. Orosz, just real quick, you mentioned Dr. Scott Eccles, his scans and stuff. We did a webinar with him, so just to remind people
oh,
that.
I love him.
Yeah, so we have a webinar that we did with Dr. Eccles on his scan technology. It's really fascinating, like you said. So.
Oh gosh, yes, yes. And see, he talks to me all the time because because in my first life I was an anatomist, a neuroanatomist, and so he's got some amazing things. I love to just talk. He doesn't know how amazing he is, because there are times that I can look at some of those brain sections and say, Scott, I can see that there's a tract here that no one's ever described. He goes, oh, really? Yes, I can. So anyway, so I'm going to get some of his stuff. Okay, you guys, I'm going to try to get a couple of his cases when we talk about this again in October. Okay, so I'm going to steal some of his images. Okay, so when we talk about clinical disease, so I'm talking about, when we talk about clinical disease, I tend to want to call it avian ganglioneuritis. And I always include it in my list of differentials. It doesn't mean that it's all that is the only problem with feather damaging behavior, or with neurologic disease, that's the only reason you have something, because you could have lead poisoning. I mean, you can have all kinds of stuff, right? So that's why I have that in there this way. Now we know, we've learned a lot about avian Bornavirus. A lot of it came out of the work of Richard Goff and Nigel Harcourt Brown, where they first said, you know, we think there are these viral particles in the feces. And so we have the work by Dr. Kistler and his group, and Hankavora and his group, and Natty Gantz, and a variety of people have worked on this. And they found that Bornavirus could be the reason for PDD, proventricular dilatation disease. So they were able to do Koch's postulate, as it's called. They could recreate Bornaviral disease with introducing avian Bornavirus. Now, I want to go hear Dr. Rossi at the meeting, the international meeting in Belgium, and he talked about something, and I'll talk to you about this here in a minute. And I had to run him down in the parking lot. Here was this American, like, you know, talking to him about things. So let's go on with this story and talk about this for a moment. So the first thing that we should all know is that this Bornavirus is a negative stranded RNA. It's an RNA virus. What does that mean to us? Well, it isn't like the virus is going to give you the common cold. RNA viruses, by and large, are not hardy. They don't last long in the environment. And so it's harder typically to pick them up. And then you see this list of viruses that are in this particular viral group that like and seek the nervous system, okay? And that's what highly neurotropic virus means. It's going to seek some part of the nervous system. And typically associated with them is that they often don't cause disease until the immune system drops, but they also are there. They're there all the time. We can't get rid of them, okay? So that's the basic commonality of these types of viruses. And you need to understand that owning your birds, that this is a virus that's going to affect the nervous system. It's once they have it, they got it. We can't get rid of it. And a lot of times they're not going to show symptoms. And when they do show symptoms, it has to do with the immune system of that particular patient, okay? So we don't know how long the incubation period is yet. There's a lot to be learned. The clinical disease and the pathology that we see is going to vary based on that patient and that immune system, because I just showed you pictures of birds with different symptoms. There was the old world birds that tend to have effects of the cerebrum, the head, or the cerebellum, and they would get the shakes and the jitteriness or seizures. Then I showed you the PDD birds, the New World species, and those birds tend to have problems with the GI tract, okay, the upper part of the GI tract. And where that happens is dependent on where that virus invades the nervous system or within the nervous system. For example, that dorsal vagal nucleus I talked about, that nucleus is this big thing, and within it are small little cell clusters. And this, let's say this little cell cluster here, it goes to the crop. And this little cell cluster here goes to the proventriculus. And this little cell cluster here goes to the ventriculus. So if it only affects the crop, that's the only thing you're going to see is a change in the crop. But if it affects all of them, the whole thing's going to have a problem. You see where I'm going with this? And many of you have asked those types of questions like, well, why does my bird show this symptom, but this other bird shows another symptom? It depends on where it was affected, where that virus started to hang out. And let's talk about that a little bit more. So then the other concern is, well, you know, what happens if my bird has Bornavirus? What do I do? What do I do? I'm like, I'm panicked. Yeah. Well, this study shows that Dr. Michael Liertz did a study in Germany. He found that just under 50% of healthy birds were positive for Bornavirus. In a sample of aviary birds with known PDD issues, 45% of them were positive. In studies at Dr. Delhausen's lab, about 35% of the birds were positive for Bornavirus. So a lot of birds can be positive, but much fewer will have signs of avian ganglioneuritis, show symptoms of disease. Does that should start, you should start understanding that.
Yeah.
So how is it transmitted? We're learning, but we don't know this. And I have my proposal and I'm still sticking to it because I had to be interrogated by Dr. Gerlach. Okay, so the standard thing that people think about is it's like a DNA virus. You suck it in your nose and you're going to get infected. Just, you know, like you've got the cold. And they've tried to do that and it doesn't work. It appears, honestly, I'm going to narrow this down and say, in my estimation, it comes to the urine and or the reproductive tract. So when you set one bird next to each other, they don't tend to become infected unless they're sitting in the same cage for long periods of time, or unless bird A eats the poop and essentially licks up some of the urine of the bird B. Okay, because a number of you asked me that. And that's because Dr. Delhausen is a fabulous guy who's done a lot of work, and he and I sent him lots of samples, and graciously we looked at them all together. And I did a group of birds in somebody's house, and one bird had it very badly, was very sick. No one else in the house got it. Okay, that's that horizontal transmission. Vertical transmission is a different thing. So when they try to put it in the nose, they just can't get anything to work. Now, I truly believe that the majority of times this is vertical transmission, and I believe that the hen is essentially the one that has to be infected from the male, and most of the time that Bornavirus is going to come through the reproductive tract, into the egg, through the embryo. So we saw, and this is because of a study that we weren't able to publish because of sensitivity of the issue, where we had about 100 citizen birds we followed, and we did a really big study, and we looked at where they came from and who got mixed with who and when did they get together and all this other stuff. And it really, really looked like that it all came from one spot where the eggs were artificial, they were pulled and they were artificially incubated. And that was a really big clue, frankly, because when we traced it back and we found that those eggs were pulled and artificially incubated, we knew that there was less of this bird to bird, but mama in the egg. At some point, as this egg is coming down the oviduct, this thing is getting infected. This embryo is going to get infected. And at some point, then they will have a problem. So in 2007, if any of you guys know Helga Gerlach, she was the German Grand Inquisitor of avian pathology. And so I answered the question really correctly. And then she said to me, Susan, it is transmitted in the vitelline membrane of the egg. I thought, oh, and I keep telling people this. I keep telling them, please look there, look there. And that's what I want the researchers to do. So Dr. Delhausen did, and he found that in Kia chicks that had neurological signs, both of the parents were Bornavirus positive. And then when he looked at eggs, 22 out of 23 eggs were positive. So that's an interesting thing. So it's important to know that Bornavirus is present in healthy birds. Remember, we talked about that infections are lifelong. Remember that group of viruses, it stays persistent. And when we see signs, it's due to some kind of stressor that alters the immune system. And one of the big ones is reproductive. Listening to Helga, my view, because we knew that Helga was always right, was that since it gets incorporated into the egg, that I think that the virus stays quiescent until the reproductive hormones go up. In general, it doesn't have to be all the time. It doesn't have to be all the time, but most of the time or often, because we don't know yet, we don't have the data yet to tell us exactly how often, but I think that most of the times this is what happens. And then that is some kind of signal that alteration of the immune system because of reproduction when the hormones go up. And then that's when the virus will want to be transmitted. But that is when we typically see symptoms, okay?
Dr. Orosz, question for you. Is there any connection, like, is it seen more in males versus females? So you're talking about hormone, is it equal? Equal?
No, so you'll see it in males and females. So you'll see it in adult males and females, and you'll see it in chicks and so it doesn't matter because that reproductive stress is still there. So then when the male is going to breed with the female, it can be transmitted then to the female. See, so I have not. Now, maybe if people look at this closer, if we get researchers to look at it more closely, there might be a dichotomy, but I don't see it clinically, okay? And again, because of the hormone status, young birds and old birds are commonly more affected. And I believe that stresses can be triggers, and particularly the hormonal season of the year when the reproductive hormones go up is a seasonal trigger. Does that make sense, Laura? Okay. Yeah. And then again, remember I called it neurotropic, which means it wants to then infect certain ganglia and certain nerve cells. Now, what I'm trying to show here is here's a nerve cell, right? And then this is the nerve cell membrane. So when this virus wants to invade this nerve cell, when it does that, it exposes these ganglioside proteins, and this is a ganglioside protein over here. You can see this ganglioside protein here, which was used to be inside. It was inside the membrane like this. And then this virus invades, and when it invades, it goes whoop, it comes out. And then that ganglioside protein, which it normally is being hidden, is now being exposed. Okay, and when it's exposed, the body goes, oh my God, what's happening? What's happening? This isn't right. It's not right. It's not right. So then we make antibodies to those gangliosides because now they're exposed. So these antibodies, that's what AB stands for, binds to these ganglioside proteins, and then you get activation of complement, and then you get activation of lymphocytes. See, this picture here that you see here of this ganglion, what you don't really see a lot of ganglion cells. Well, if you understood the histology, you don't see a lot of them, but you see a whole bunch of lymphocytes, because this ganglia is being invaded by lymphocytes because those ganglioside proteins are being exposed. And then the alarm bells go off, ding, 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 fire over here in ganglion number 1. And so the lymphocytes go rushing in. And that's at that point, when the lymphocytes go in there, then it's like, that's when the symptoms occur, okay? So it's the lymphocyte invasion that causes this problem. So it really is an autoimmune disease. And we can see this, and I'm kind of getting ahead of myself just a tiny bit, because of this thing called molecular mimicry. In other words, this exposure of these ganglioside proteins doesn't just occur with Bornavirus. It can occur with infections of Campylobacter, Mycoplasma, Haemophilus, influenza A, and paramyxovirus. And they all can produce a ganglioneuritis. And so we have this cross reactivity that occurs. So the gangliosides that are causing this, and there are a variety of them, and I have them listed. Those yellow ones down there are the different ganglioside, GM1, GM2, GM3. Those are different gangliosides. And different gangliosides tend to be found in different parts of the brain. And these different viruses or bacteria tend to want to seek different gangliosides. Hence the difference in the symptoms. Okay, so these gangliosides are kind of the key to the puzzle and their exposure from the immune system. Now here comes the next big dilemma, and then I'm going to get back to what we're talking about here. And just this is our dilemma. Our dilemma is, as veterinarians, we see birds that have, in the past, that have classic symptoms of PDD. Man, they got poop. Their poop has seeds in it. They have wasting. They have an enlarged proventriculus, and we go, oh, okay, we're going to send it out. We're going to give them a Bornavirus test, and it comes back negative, right? Now, there are a lot of reasons that it can come back negative. One is that you didn't do the test properly, you didn't do it the right time when it was circulating. I mean, there are those things. But let's pretend for just a second, with the work that Dr. Rossi did, that he found a bunch of birds that, man, they were classic ho-hummer. They had it. And in fact, what he did is he did necropsies. He took blood. He took blood from those birds at necropsy, and he tested them. They were negative, but they had the histologic features of Bornavirus, of this PDD symptoms. And they said, well, how do we deal with that?
Yeah.
Okay, so what he did, as he said, well, maybe it's not just the virus. And I've already kind of suggested that to you. What if we take purified ganglioside, take the ganglioside proteins, and we inject them into cockatiels, but we don't give them Bornavirus. We just give them the ganglioside protein. And we put it either in their coeloms, which is what this picture shows. We're either going to give it to them orally or in their eye, an intraocular, or we're going to put it in their coelomic space, their abdomens. And we're going to see what happens. Well, this is what happened. A month later, 100% of the cockatiels, all three of them, that were given it, where they put it in their intestinal space, that abdominal space, and 33%, a third of those that had been given it orally of just the ganglioside, again, they developed signs similar to what we described as PDD. When he looked at them histologically, they were what the pathologist classically describes as having PDD, this lymphocytic plasmacytic ganglioneuritis in those birds. So they had PDD, but they never had Bornavirus. Okay, so now we know that it's the ganglioside, the exposure of the ganglioside, that is the most important thing. Okay, so this kind of shows you a bird with a huge proventriculus, which was one of these study birds. This was the histopathology of it. And again, you see, you all know now those blue dots are those lymphocytes, right? And that's that lymphocytic invasion. And those lymphocytes are T cells. They're T lymphocytes. And then here we show the macrophages in there. He's got labeled, and he's got, this labeling of this particular CD8 type thing. So this is what Dr. Rossi found. So this is when I ran him down in the parking lot in Brussels. Like, listen, I got to talk to you. And like, he didn't know a lot of English back then, and I certainly didn't know any Italian. And I knew this guy was really knew his neuro, because when I would tell him, when I'd ask him different ganglia in the brain, he could tell me yes or no, he could find this. I knew he was on to something. Okay, that's when I knew something was there. So in terms of diagnostic testing, I don't know how we are doing on time, but we're having so much fun.
Oh, we're doing good.
So, okay. So there is real-time PCR for Bornavirus. We do know, and Dr. Delhausen is the master of doing it in the blood. Nobody else could figure out how to do it, but he did. And so he had to use a very special technique to do it. And I have to tell you, I sent him lots and lots of samples. And that the Bornavirus is going to be shed intermittently because, again, stressors and reproductive hormones. So he had, and this is his whole thing, he had to have a fresh sample. It's got to be sent in a certain period of time. And you all know that because those of you who were back in the early days of this, when Dr. Goff talked about this, they would find these, what he called viral ghosts in their droppings. I'm going to call it droppings because it wasn't just the fecal. And we didn't realize, we should have looked at, was it in the fecal or was it in the urine? That's what we should have done, but we didn't do that. And I'll bet you it was just in the urine. Okay. And those ghosts, if I slammed them into the EM scope at the University of Tennessee in the morning, and I looked at them by the afternoon, I could see them. But you couldn't see them by the next day. Okay. Again, remember I told you that it was an RNA virus. It doesn't last long in the environment. Remember that. Okay. So again, positive samples. We could do this with Bornavirus. We found that different labs have been able to do that. And then what about false positives, false negatives? False positives, hardly ever. Hardly ever, very uncommon, be very difficult to do. False negatives are going to be common because A, you didn't send the sample right, you didn't send the right sample, you let it sit in when it got hot, because this is a finicky little virus, and you got to be careful. That's why it isn't shed in a horizontal way, which causes a lot of disease. That's why I think that. Okay, so all of you are concerned about worldview number one. I can tell that when I saw the questions. Oh, my bird is positive for Bornavirus. It's going to have PDD. It's going to die. It's really bad. It's like awful. Like I'm going to kill myself because my bird is positive for Bornavirus. No, it might, depending on the bird and its immune system. But I just showed you a graph, Michael Lurtz in Germany, just under 50% of birds are positive for Bornavirus, and they look fine. They're looking fine. And then in Dr. Dahlhausen's lab, about 35% of them test positive. Most of them look fine, okay? So they can have Bornavirus, but they don't have to have disease. Now, the flip side is some of them who have Bornavirus have really bad disease and some of them will die. But just because they're positive for Bornavirus doesn't mean that they are, okay? And then the other questions that people had is this anti-ganglioside antibody assay that was developed by Dr. Rossi in Italy, and Dr. Dahlhausen was able to buy the rights to this so that we could have it available for us. And so, as you could see, that when you have disease, when you have neurologic signs, that's one of the things that you want to do, is you want to consider running this anti-ganglioside antibody test, because your concern as the owner and the clinician is could avian Bornavirus be a problem? And you just found out, yes, it might or it might not. And there might be other things that are causing these gangliosides to become a problem, right? We had that list of other things due to molecular mimicry. And so these anti-ganglioside antibody assay will help us during this phase of neurologic symptoms. Now, that thing's going to fade too, just like in time, the Bornaviral, the avian Bornavirus test might be negative because it might not be floating around in the bloodstream, okay? So in the work that Dr. Rossi did, 98% of birds with clinical disease, this bird has some clinical disease. It had elevated anti-ganglioside antibody levels. And that happened regardless of if they're positive for Bornavirus, okay? Now, we have a few minutes to talk about treatment. And this is also important. I told you, and you saw here, that there's a whole bunch of T lymphocytes sitting there. If you want to, they're essentially glunking up. I'm using these really great, anatomic terms. Glunking up the nerve conduction with those T lymphocytes hanging out there in that ganglia. Well, we have Celebrex. It's a COX-2 specific inhibitor, which means it selects for and will concentrate in nervous tissue. So this was the old go-to for me to use, and I use, it says it gives a range here, and I use 20 mg per kg twice a day, and that's pretty darn close to the recent research out of Belgium, where they've done some pharmacodynamic studies and pharmacokinetic studies of Celebrex or celecoxib. In addition, there's also robenacoxib, which is under the trade name Onsior, and I have only used it by injection. And this is between 2 and 10 mg per kg. It can be given IM, and it depends on the metabolism of that bird. I found that sometimes you can only, about every 3 days, you have to give it in these small conures, up to a week in some of these bigger birds, and even in chickens. And then that's when you're going to treat, you want to treat IM. If the GI tract's not moving, why put a drug in it? It's not going to work. You got to give it IM, okay? Now, the other one that a lot of people tend to use, because this was our first non-steroidal anti-inflammatory, is meloxicam, but it does not select as well for the central nervous system as the coxibs, the Celebrex or the Onsior, okay? And in a study done by Sharman Hoppus, she found that giving this drug to cockatiels made them worse when they were given Bornavirus and they had symptoms. Now, it could have been in part because the dosing was low, okay? And that could be part of it, but I'm gonna tell you, having done this for as long as I have, you wanna use either robenacoxib or you wanna use Celebrex. Now, in terms of the neuritis issues, this potential for shooting pain, this is gabapentin. I tend to use it, start at 50 mg per kg twice a day. This can be pain shooting down the legs, pain in the spinal cord to the wings. So it can also be used for seizure control. And another one that we use with seizure control is Keppra. So those are two of the drugs that might be used. And then again, we had the New World species, cranial nerve 10, dorsal vagal nucleus, affects the proximal part of the GI tract. So you have a slowdown of the crop, the proventriculus, the gizzard, depending on where that virus hit in that dorsal vagal nucleus. And when you have bacterial overgrowth, you get Clostridium, you get anaerobes, you get gas. So you've got to get rid of that. You've got to take care of your patient. So you use things like Clavamox. You use things like spectrinomycin if they're in better shape. For the yeast, you might consider nystatin. And in a study done by Dr. Leertz, he also found that most birds that have avian gastric yeast, which are these long, or the mega bacteria, as Helby used to call them, these big, huge yeasts that look like huge rods, 70% of those birds that had this also were positive for Bornavirus, because there's an alteration of the normal motility of the GI tract. So we've got to treat those yeasts, and we got to speed the GI tract up. And we got to reduce the inflammation in the GI tract. So that's where I try to use things like your Celebrex, your robenacoxib. Then you can think of things to reduce that nausea and speed things up using ginger if you want to go in a non-traditional way. I tend to think about using metoclopramide, but you got to be careful of the dosing, and cisapride. Those are all things that we're going to use to try to speed that GI tract up and reduce inflammation. Now, I told you before that one of the things that I am concerned about when I always talk about this is, hey, before your bird started having symptoms, did it start acting hormonal? What was it doing right before this? And typically the owner is going to tell me, yes, that's going to happen. So we think about things to reduce that hormonal stressor, to reduce that turnover of the viral particles in those ganglia, and I give them desirone implant. Typically I don't. I tend to use Depolupron in those birds. And then depending how bad they are, we need to think about gavage feeding. And I helped in their original design of the critical care diet from the Lafeber company. And this is not an infomercial, but let me tell you something. It is truly the one that's close to being an elemental diet. If you need an elemental diet, don't screw around with something else. You got to use this diet that Dr. Clason actually designed, because it has diatripeptides, isoamino acids, easily digested fats, high levels of energy. And when you have a bird that isn't absorbing right because its GI tract isn't working properly, you really have to use that drug. Okay, then somebody asked me, and I'll just kind of end here, you guys, because we only have a minute or so left, is there was the study that Dr. Rossi did using the mycobacterium intradermal injections of essentially BCG in the United States. And he combined that with robenacoxib. Really a great idea, okay? But, and this is kind of what he did. He found he had these healthy parrots, but then he went and he had birds that were unhealthy, and he looked at their gizzard, and then in the affected bird, you've got this, you know, T cell lymphocyte invasion, and then he gave them the BCG intradermally in the skin, but he also gave them robenacoxib. And then when he did that, he essentially tricked the immune system into going and attacking the area where the BCG was administered, and it took them away, see, from the area in the gizzard and that treated parrot. So it reduced the glunking up, as I describe it, of the nerves and the ganglia, okay? So he got a lot of birds that worked. Here's the problem. Can't use it in the United States of America. For those of you who are listening in other countries, you'll probably be able to use this technique. Really a great idea. So in closing, you need to understand that avian bornavirus fluctuates. It can stay dormant, then during the reproductive period or during high stress periods, that's when it gets activated. And that's when you get this immune suppression that happens, you get T lymphocyte invasions, and you have changes also during the reproductive period. That's when I think most of this happens. And that it's an immune mediated disease because we expose those glycoproteins, the ganglioside proteins, and we get this response. And this can be induced not only by Bornavirus, but this exposure of these ganglioside proteins can also be by other types of neurotropic bacteria and viruses with this condition called molecular mimicry. And also, you can have Bornavirus without any disease. So that's an important thing to remember. And I'm basically out of time, but by God, this was a fun hour, wasn't it? We learned a lot.
Yeah, absolutely.
And I don't want to confuse you, you know, but I wanted you to really understand this as we know it now. Okay, and we're going to, you know, then we can answer more things in October. We'll take those questions and try to go through this. But I wanted to set the stage for you. I thought that was really important since we only had an hour. And you could see, I could talk about this for days, right? Days. Yeah.
Wow, that was insanely fascinating. And we have we do have a bunch of really good questions lined up. So when we come back in October with you and Dr. Dahlhausen, this will be like, this is a bunch of thank you. So that was an awesome presentation. Totally appreciate that, walking us through that. That's a lot of material.
And thank all of you for listening, right? You know, to take the time to listen. And for those of you who are purists, right? I know that I kind of oversimplified stuff, but that's okay.
Yes.
Because we all have to understand it.
Yes. And I do want to remind everybody who's tuned in is these webinars are recorded and you can watch them. If you go to the YouTube channel, type in Lafeber Pet Bird and you'll see these webinars pop up. So you can rewatch this one. You can rewatch the ones before, like we mentioned, Scott Eccles' scan webinar. You can watch all those. They are recorded. So, and just really quick, I wanted to give, we do have our giveaway winner today for the AV cakes. And that is, let me see here. Let me go back up and then it is, where is my little note here? I got all these thank yous pouring in. So I'm looking for the webinar again. Let's see here. Sorry, I just they're going to get a packet. They're going to get a bag of the Avi cakes as well as a food of their bird's choice, other Lafeber product. Judith C. Congratulations, Judith. Hopefully that makes your weekend and your bird's weekend. Dr. Orosz, really appreciate this. Very fascinating information.
Thank
Very
you.
important.
Thank you all. And thank you for taking the time to really hone in and listen. It was great. I had a great time. I hope you did too.
Absolutely. And just to remind everybody real quick, we don't have a webinar this the first Friday in August, but we do have one the second Friday. So join us then. And in the meantime, I wanted to wish everyone a fabulous weekend. Everyone stay safe and all the best to you and your flock. And thanks again. Awesome webinar. Appreciate it.
Thank you.
Bye.